Hi, everybody. Welcome to our lecture on Chapter 8, Cell Transport. You can see in the animation here, there's obviously something moving, and the question is, what am I? A good answer might be like beating heart cells, but that's wrong. It's something a little more sophisticated, frankly, than just beating heart cells. These these are cells, and they're obviously moving some stuff around. Where do they belong? Well, those are actually structures called chromatophores, and you find them in squid and the mollusks and other creatures like a chameleon. They can move pigments around to change their color. And so this is an example of cell transport, a little more sophisticated than the one we're going to cover, but I thought it was an interesting picture. And so anyway, remember that we talked about the 7 characteristics of life when we learn about biology, and it's charge them, and there's 2 H's. One of the H's is homeostasis. When you think about homeostasis, I want you to think about cell transport. So remember that it's the maintenance of a stable internal environment, even when the environment is changing, and we maintain homeostasis largely through the work of the cell membrane. So we've spoken quite a bit now about the cell membrane. You should recognize this molecule at the top. It's called a phospholipid. Now remember, it's got 2 fatty acids. One fatty acid is saturated, the other one is unsaturated. You can see it's got a little kink in its tail. Those fatty acids are hydrocarbon chains. They carry no charge, therefore they are nonpolar. And we all know that fats and water don't mix. It's nonpolar, it's hydrophobic, it hates water. On the other hand, you'll notice that glycerol molecule is attached to a phosphate group as well. So phosphate is sort of abbreviated as the PO3 minus group. Phosphate has a negative charge. Since it's got a charge on it, it's polar and it loves water. Therefore, we say it's hydrophilic. And because all cells exist in a watery environment, and all cells have a watery interior cytosol, it creates this lipid bilayer. And hopefully you're familiar with this by now. Only a few substances can pass directly through the lipid bilayer, and they need to be nonpolar and small. For example, gases like CO2 and oxygen will readily diffuse right through the cell membrane. Now, diffusion is a special concept. It's the movement of material from an area of high concentration to low concentration. That's going to be a central theme in our discussion here. If you're big, you need to be transported. If you're polar, you need to be transported. So, how do large substances like glucose get across the cell membrane? How does water pass through the cell membrane? Well, it's the work of these intrinsic membrane proteins. Intrinsic means integral to, or central to, or within. And so the intrinsic membrane proteins are embedded in the phospholipid bilayer, and they have lots of different roles. Some are receptors. They bind signals like hormones. Some are carrier proteins. They're pores or channels. You can see in the bottom diagram, there is an aquaporin. And hopefully you recognize those little tiny red and blue molecules as water. That's right. This is how water passes through the cell membrane, through a intrinsic membrane protein called the aquaporin. We also have enzymes embedded in the cell membrane to break down certain substances or move certain substances. Remember that enzymes catalyze chemical reactions. We have CAMs, or surface adhesion molecules. For your tissues, you definitely want them to stick together. And so there are cellular adhesion molecules that connect cells adjacent to one another in a strong mechanical way. We also have cell surface markers, and these guys typically are glycoproteins. What's a glycoprotein? Well, glyco refers to glycogen. These are long chains of glucose, and protein is simply that. So I have proteins with long chains of glucose, and you can tell a lot about a cell. These are identity markers. They say, hey, it's me, I'm not a bacterial cell, and you can even tell how old a cell is by how long those chains are. Of course, there's also cholesterol embedded in the cell membrane, and that makes the membrane more or less rigid. Well, again, we're going to talk a little bit about thermodynamics here. Remember that this is the study of how matter and energy interact with one another in chemistry specifically. And thermodynamics really drives cell transport in a big way. Remember, the first law of thermodynamics is that matter is not created or destroyed in any change in the universe. However, every time there is a transition, every time there's a chemical reaction, energy is lost to the universe as heat. The second law of thermodynamics says that the nature of the universe is to become more randomly ordered. That is more disorderly. And we call that property entropy, and it's always increasing. Of course, for a minute, you guys are a little more organized than you might be before. So cell transport is all about considerations of the first and second law of thermodynamics. And in that sense, we divide cell transport into two types, active and passive transport. Passive transport is easy to identify. No energy is used, and material moves down the gradient. You can see this boulder just move down the gradient there. On the other hand, active transport always requires energy, and it moves substances up their concentration gradient. So again, passive transport requires us to understand these ideas of concentration gradients and equilibrium. Remember that if I lit a fire in the corner of the room, initially the smoke particles would be concentrated in that area. But if I left them room for a while, they would be equally distributed. Initially, I have a concentration gradient. After some time zero, that system would go to equilibrium. And that's what we're talking about when we say that substances move down the concentration gradient. Just like this bike rider. The question is, should he be wearing a helmet or a hat? That's called neuronal posturing. You don't want to do that. Well, we've already given you a couple examples of simple diffusion. Small nonpolar molecules like the gases CO2 and oxygen can just pass right through the cell membrane. Now, there are other substances that can pass through the cell membrane, like steroid hormones, right? They're fatty, so I could put a little testosterone on your skin and it'll absorb right through. Down at the bottom of this animation, you can see that the nature of stuff is to go to equilibrium. So if I put a bunch of orange molecules in that solution, they will move to equilibrium state. And we say, by definition, substances move from areas of high concentration to low concentration. That's the definition of diffusion. Well, if you can't simply diffuse, then you can be helped along. And that's where carrier-facilitated diffusion comes in. To facilitate means to help, and we're helped with carriers. A couple different types of carrier molecules include channel proteins and carrier proteins. Channel proteins are just that. They're sometimes called pores, and they can let things like ions or water pass through. They have interior amino acid sequences that complement whatever ions there. So what I mean is this. So remember when we talked about amino acids, they can be positively or negatively charged. Well, if I'm an ion channel for sodium, that positively charged ion, then the interior amino acid residues will all be negatively charged, and that will actually invite that positively charged ion through there. For larger molecules, we have carrier proteins. And so, for instance, we talk about glucose. Remember, glucose is very large and very polar. How do I get it across? Well, I use a carrier protein. Here's some illustrations of an ionic pore and an ionic carrier. At the top, you see the glucose uniporter. The glucose uniporter is perfectly designed to bind glucose and allow it to pass through. In a way, it's a gated channel. Once it binds glucose, it can actually push it through, like you see in the diagram to the right. The potassium channel is really pretty simple. It's just an open pore and positively charged. It's perfectly sized for potassium to pass through, and that's it. So remember, for examples for carrier proteins include the ionic pore, which can include the aquaphorine as well, a potassium channel, sodium channel, and then for carrier-facilitated diffusion, we think of the glucose uniporter. Another example of a passive cell transport is osmosis. Now, osmosis, simply stated, is simply the diffusion of water. Sometimes the diffusion of water through a selectively permeable membrane. And so remember, cells exist in a watery environment. The interior of the cell is a watery environment. So the question is, what would happen to you if I put you in the ocean for 3 days? What would happen? And the answer is not good things. There's a lot of problems with being thrown in the ocean for 3 days. Fish bites, thirst, etc. But in large part, the real issue has to do with something called the osmotic pressure. The ocean is really salty. Your cells are salty too, but the ocean's probably more salty. So now we can think about not just salt concentration, but what about water concentration? Now that's where the idea of tonicity emerges. So I know it's salty in the ocean. I know that my cells have dissolved solids as well, but which one has more? And then the question becomes, where is the water concentration highest? Is the water concentration highest in the ocean, or is the water concentration higher in my cells? And the answer is the ocean. I'm sorry, no, other way around. The answer is your cells. So where's the water want to go? Well, the water's going to go, just like every other substance, the water will diffuse to the area of lower concentration. So think about it again. If there's a lot of salt in the ocean and there's less salt in your cells, where does the water in your cells want to go? Well, it wants to go out. That's why you can't drink seawater. If you drink seawater, you might feel good for a minute, but then what's going to happen to the cells in your stomach? Well, the cells in your stomach are going to be up against really salty water. And the water inside of those cells is going to want to go out, and it will. And in fact, I think typically what happens when you drink seawater is you vomit, and not only that, but you become more dehydrated as the water leaves your GI tract. So anyway, this brings up these terms: hypertonic, hypotonic, and isotonic. And these are relative terms. So we talk about two conditions as either being hypotonic or a system that's hypotonic with respect to a hypertonic system. So to answer this difficult concept, I want you to think about, how do I make a pickle? Well, you make a pickle by taking a cucumber and putting it in salty water. So where is there more salt? In the bucket of salt water you put it into, or inside the cucumber? And the answer is that it's saltier outside. Therefore, the water concentration is lower outside. Therefore, where does the water in the cucumber cells want to go? Well, it wants to go out. And of course, that causes that cucumber to shrivel up into a pickle. That's a simple idea. What if I took that cucumber and threw it into a bucket of water? Well, pure water has a very high water concentration. The cucumber obviously has some dissolved solids in its interior, so it's hypertonic with respect to the exterior. And so that water from the bucket will want to go into the cucumber and it'll swell up. Ay, ay, ay.